Can I go? I have to put some text close slides. Good afternoon, everyone. Welcome to today's research conference. In the interest of time, while Dr. Kurtzberg's talk is being loaded up and she's getting prepared, I really wanted to introduce her to this group. So just a quick show of hands. How many people are familiar with Dr. Kurtzberg's work? A lot of fans. Okay. For those of the people who didn't raise their hands, sit next to somebody who did raise their hands because you're going to need help getting your jaw off the floor. Her work is just phenomenal, and it's a real pleasure to have her present to us today. The title is aptly named Game Changers Using Cord Blood to Help the Brain. Dr. Kurtzberg is the director of the Pediatric Blood and Marrow Transplant Program here at Duke. She's also the Chief Scientific and Medical Officer of the Robertson Clinical and Translational Cell Therapy Program, the director of the Carolinas Cord Blood Bank that she started, and the co-director of the Stem Cell Laboratory. She's also the Jerome Harris Distinguished Professor of Pediatrics. She joined Duke back in 1983 after a few years of a research fellowship here, and since then has had a meteoric rise and built a phenomenal, innovative program in stem cell therapy, umbilical cord blood banking and transplantation, with a variety of novel applications of cord blood in the emerging fields of cellular therapy and regenerative medicine. She has pioneered the use of umbilical cord blood as an alternative stem cell source for unrelated hematopoietic stem cell transplantation. Several years ago, she developed the Julian Robertson Cell and Translational Therapy Program here at Duke that focuses on the translational studies from bench to bedside with a focus on bringing cellular therapies in regenerative medicine to the clinic. Several areas of application have included neonatal brain injury and cerebral palsy, as well as preclinical studies in a variety of conditions, acquired and genetic conditions across the clinical spectrum of pediatric disease, including some innovative work in young children with autism spectrum disorder. She established one of the largest unrelated donor cord blood bank programs in the country, the Carolinas Cord Blood Bank, in 1998. It has a current inventory of greater than 40,000 units and has provided cord blood units to over 2,200 patients. In addition, her lab has developed novel assays to predict cord blood potency from segments attached to cryopreserved cord blood units and is performing translational research testing cord blood expansion, as well as a number of applications in tissue repair and regeneration. In 2012, she also helped the Carolinas Cord Blood Bank receive FDA approval for DuCord, a stem cell product derived from umbilical cord blood for use in transplants between unrelated donors and recipients. She holds several INDs, has published over 300 manuscripts, chapters, and scientific reviews, is on a number of international, national, and local organizations to facilitate broader application of blood and marrow transplantation. Her work has been recognized by a number of bodies, including the Lifetime Achievement Award from the Pediatric Blood and Marrow Transplant Consortium in 2012. Please join me in welcoming Dr. Kurtzberg.
Thank you, everybody. It's a pleasure to be here and to get the opportunity to tell you about our work. And I'll say at the beginning and I'll say, excuse me, at the end that the work I'm going to present really represents efforts of hundreds of people over more than 2 decades, not just me, but I get to tell you about it. And today I'm going to talk about using cord blood to treat both genetic and acquired diseases of the brain. Danny Laskowitz gave me this slide, but I think it's a good way to start. Just a few comments about how the cord blood field started. Back in 1988, this little boy who is named Matthew and was a patient at Duke had Fanconi anemia. His bone marrow was failing. He needed a transplant, which is really life-saving in that disease. His mother happened to be pregnant with a baby, and we tested that baby in utero and found that she was healthy and not affected with Fanconi, which is a genetic disease, and also that she was a match to her brother. A number of investigators were working in the day studying whether cord blood contained hematopoietic stem cells and progenitor cells, and it looked enriched for those cells. And a group from New York got together with us, and we approached the family and asked them if they'd be willing to let Matthew have a transplant using his baby sister's cord blood as opposed to her bone marrow. We knew that her bone marrow could be a backup if the cord blood didn't work. The family said yes, and we sent them all to France, where Elian Gluckman, shown in this picture, performed the transplant. Dr. Gluckman really was the pioneer in figuring out how to give patients with Fanconi anemia appropriate chemotherapy and get them through a transplant. And so this family that lived in Salisbury, North Carolina, had never been out of the state, all went to France and lived there for 6 months while he had his transplant. And you can see these were early days where they used sort of makeshift laminar flow behind curtains. The cord blood was collected in a bottle when the baby was delivered with some heparin, and it was mixed with DMSO and just frozen in big bags that were the cryopreservation bags people used in research freezers. Hal Broxmeyer took the cord blood in this dry shipper on an airplane, bought an extra ticket for the seat next to him, and brought it to France where it was thawed and infused. And Matthew, who's now 27 years later, 33 years old almost, is healthy, well, and grafted with his baby sister's cord blood, still comes to Duke for checkups, is taller than me, which is one of our patients' goals, and is healthy, fortunately. And he really was the first experiment in a child, in man, that proved that cord blood contained enough stem cells to repopulate the blood system and to durably engraft the blood for now decades. I always say that if we were starting today, I don't think we could do this. The regulations are very different, and we would have had to do preclinical and animal studies, which were not really done before this transplant was done. And in a way, it may be good luck that this happened 27 years ago. But his transplant paved the way for the field. And since that first transplant, again in 1988 in France, with a matched sibling, there were other transplants done between matched siblings over the next few years, showing that the main complication of bone marrow transplantation, which is called graft versus host disease, where the donor cells attack the patient, was much less when you used cord blood as the graft source as opposed to bone marrow. And that's probably because cord blood comes from a baby that has just gone through pregnancy, and where tolerance is induced in an immunologic sense between the mother and the baby, who are not matched to each other. They're half matches to each other, but they tolerate each other during pregnancy, and that property is carried over to cord blood cells. And that gave people the idea that maybe cord blood could be used in the unrelated setting, and maybe it didn't have to match completely, which is always a problem for up to half the people who need a donor for transplant. So Pablo Rubenstein at the New York Blood Center started the first unrelated donor cord blood bank in the US in 1992 with a grant from the National Heart, Lung, and Blood Institute. And we had started the pediatric transplant program here at Duke in 1990. And we partnered with Dr. Rubenstein to be a user of the units from his bank. And we performed the first unrelated transplant ever in the world in a little boy with leukemia in 1993. We also were fortunate enough to establish our cord blood bank through a grant from NIH in 1997. That's called the Carolinas Cord Blood Bank. And we, as you heard, have built up a large inventory of public units for distribution for transplant. Now, if you fast forward to today, there have been more than 35,000 cord blood transplants in the unrelated donor sense performed around the world. There are more than 160 banks. The public inventory in the United States is estimated to be at about 220,000, and worldwide it's somewhere between 700 and 800,000 units. The private inventory, so private banks are commercial entities that parents can pay to store their child's cord blood for their use by their family, and the private inventory is estimated to be more than 4 million units worldwide. In 2005, we were fortunate enough to facilitate legislation through the U.S. Congress, which established the C.W. Bill Young Transplant Program, which brought together the National Marrow Donor Program's adult registry and the cord blood bank's inventory into a single point of access registry, an outcomes database, coordinating centers, and a banking network. And as you heard, FDA decided to regulate cord blood as the first hematopoietic stem cell product to need licensure and issued final guidance in 2011. And the cord blood bank at Duke got its license in 2012. Currently, it's estimated that there are about 5,000 cord blood transplants performed annually around the world. Now, today, I'm going to talk about using cord blood to treat genetic and acquired diseases of the brain. I'm going to start with genetic diseases because that's where the field started. We had been using cord blood as an alternative donor for people who didn't have a match in their family or in the adult registry for transplant. And in pediatrics, we also, besides transplanting children with leukemia and other malignancies, we use hematopoietic stem cells to correct inborn errors of metabolism. And essentially, it's a kind of cellular gene therapy. You can use transplantation as a way to permanently engraft donor cells in a person's body, and then those donor cells can provide whatever is missing in certain genetic diseases. So in inborn errors of metabolism, it's usually an enzyme, and basically the bad news is that the patient has to have chemotherapy to wipe out or ablate their hematopoietic or blood system and immune system. Otherwise, they reject the graft. So unlike a patient with cancer who might need chemotherapy or radiation to cure their disease, these patients are getting the chemotherapy only so that they can durably engraft with donor cells. But once that happens, the donor cells produce the enzyme that they're missing. It circulates through their blood. And even more importantly, the cells will migrate into the brain and permanently engraft in the brain and replace the enzyme in the brain. And through these children's treatments, we've also learned that cord blood has cells that are not of the hematopoietic lineage and can engraft in other tissues. I'm not going to really show you that data today, but cells have been demonstrated in the pancreas, in heart muscle, in skeletal muscle, in the liver, and in other organs. Now, over the years, we've treated over 400 children with these inborn errors of metabolism or inherited metabolic diseases. The common ones are listed here. They're all very rare, but if you come on our transplant unit, you think they're pretty common because a lot of patients with these rare disorders come to Duke for treatment. And all of these diseases in one way or another affect the brain. They're all lethal in infancy or early childhood without transplantation. And for most of them, there are no alternative therapies, although there are inroads being made to gene therapy for some of these diseases. All of the children, as I mentioned, have to undergo treatment with high-dose chemotherapy. We use busulfan and Cytoxan over 9 days with an immune-suppressing drug called ATG. They all receive GVHD prophylaxis for up to about a year. They all receive very expensive supportive care with different antibiotics and transfusions and antivirals, antifungals, IV feeding, transfusions, and many other things that are given to keep them alive while they go through and experience the side effects of the chemotherapy that's necessary to correct their condition. The average length of stay for a patient coming for this treatment at Duke is 53 days in the hospital on the Peds bone marrow unit, which is on 5200, which is a HEPA-filtered semi-isolation facility. But the families stay here with their children for 6 to 12 months while they get care post-transplant. And it really takes, even in an uncomplicated procedure, 1 to 2 years before the children fully recover. We learned early on that the child's performance status, or really disease state at the time of transplant, was a very important predictor of outcome. So this is 11-year survival of a group of children with inherited metabolic diseases that we've transplanted. And the red line shows you children who had a good performance status measured by a test called the Lansky of 80% or more at the time of transplant versus children who had lower performance in the green line. And what you can see is that the survival for children with low performance, which really means already progressive disease of whatever their underlying condition is, have poor survival. What you can't see in the green line is that they not only have poor survival, but they are very severely neurologically impaired, so they have very limited function. Whereas the children in the red line both have better survival and better function over time. Because we know this now, children who are in the green line are not considered candidates for transplant, and we're looking for alternative approaches to treat them. It is unfortunate, though, that many of the children who get picked up with these diseases are in families that don't know they have a risk and don't learn that their child is affected until they have advanced disease, and then they can't be treated. I'll just show you some examples of outcomes. So this is a child with Hurler Syndrome. The child in the picture in the upper left is an untreated child. You can see that his face is atypical, his forehead is prominent, his eyes are wide-spaced, his nose is flat, his tongue is big, his ears are low-set, and although you can't see it, he has this big liver, big spleen, back deformities, hip deformities. Without treatment, this child will die at 5 to 6 years of age of cardiac ischemia, because in Hurler syndrome, you build up a kind of chemical called a glycosaminoglycans, and it builds up in the coronary arteries. But these children also die with severe mental retardation and other problems. If these children are transplanted under the age of 2, they do well. About 90% survive. And this is just showing you data we published years ago in the New England Journal of Medicine, and it looks at the cognitive development in the dotted black line of the first consecutive 20 severe phenotype Hurler syndrome patients we've transplanted at Duke. And basically, this is months of age in the x-axis, and then developmental quotient on the y-axis. The red line is normal, typically developing children with the gray arrow bars. The Hurler kids are falling off the curve when they get transplanted, which in this group was a median age of 16 months. But over time, they catch up, and their IQ goes into the normal range, and they have improved function. And we have children now who are 10 years beyond this point and have maintained this improvement. So we don't know if this is merely, if you will, enzyme replacement in the brain, or if there's some rescue of brain cells from the hematopoietic cells from the cord blood transplant, but it's a good outcome. We also know in Krabbe disease, which is a deficiency of galactocerebrosidase, which is an enzyme you need to myelinate neurons, that if children are treated, and the red line is 13-year survival for children treated in the first month of life, they do well. They will have varying degrees of disability, but their survival and quality of life is good. But again, children in the green line, which are children who are presenting with symptoms even as young as 3 or 4 months of age, will not do well and will have progressive impairment, poor survival, and poor quality of life. Because of the knowledge of this, and because a football player named Jim Kelly had a son named Hunter with Krabbe, he lives in Buffalo, and was able to convince the New York State Legislature to fund a pilot newborn screening program for Krabbe disease, which has been operative for 5 years now, and there are other states implementing newborn screening for Krabbe. So this is a child who was first picked up in the New York State screening program and came to Duke for a transplant. It's very difficult to coordinate this newborn screening program because the transplant needs to be done so early. So essentially, the New York State screening lab does a screen on a enzyme screen on a dot of blood on a filter paper. And then if it's positive, they do sequencing of the GALC gene in 48 hours. That's the gene that's affected in this disease. If they see a mutation, they bring the baby back and retest to confirm. And then if the test is positive, they refer. The parents see a normal-looking baby, and they're told you have the option of doing a transplant, which has about a 5% mortality in newborns. And if you don't do it now, your baby will have progressive disease. And the parent looking at that baby doesn't know that, and it's a lot to really digest. So newborn screening isn't perfect, but that's the way it is. Now, I'll also tell you that in New York State, that has screened over 2.5 million babies. They've picked up 5 cases of the early infantile form. So this is rarer than they expected. They thought it was about 1 in 100,000. It's looking less than that. And there are a lot of issues about priorities and sort of resource allocation. But that's the state of that field right now. Now, some newborns with Krabbe still, even with transplant at an early age, have neurologic impairment. Now, these are extremes, but these are brothers. The little boy who's standing was transplanted at 3 weeks of age. The child in the stroller was transplanted at 13 months of age. You might say to me, how did that happen? But it's because their 2.5-year-old brother was diagnosed when he died of Krabbe. And then they went back and tested the family. And these kids were at the ages I spoke. So the child in the stroller had, till 8 months of age, been able to stand and cruise and babble and communicate pretty well. But then he lost those functions. And so you can see that when you transplant a symptomatic child, they do not regain motor disability that they've lost, although cognitively they're normal. Now, as we further explored this, we learned that some Krabbe babies, even at 2 days of age, have abnormal corticospinal tracts. So this is fiber tracking through MRI of the corticospinal tract of 2 different 2-day-old babies with known Krabbe whose siblings had died of Krabbe. And I'm just showing you the kind of spectrum, but this child has a normal tract. And this child is 12 now, and she's normal. She does gymnastics. She's cognitively normal. She really has no perceptible impairment. And then you have a child like this where even at 2 days of age, the tract is already skinny and ratty and abnormal, and that child doesn't walk. So we have a way now both to help parents with understanding what the prognosis of their child might be, and also a target for cell therapy, which we pursued. So in one of the children with Krabbe who died, the parents let us study her brain. So this is a section of cortex from a little girl who had symptomatic Krabbe, was transplanted at 10 months of age, and died at 20 months of age of aspiration pneumonia. And when we studied her brain, this is just a section that's representative. We stained it with DAPI, which makes the nuclei Duke blue, and then came back with XY fish to see the sex of the cells. And this little girl had received a transplant from a male cord blood unit. And you can see the green dots. Those are Y chromosomes scattered throughout the cells of this child's brain, indicating that within 10 months, there had been significant engraftment of male donor cells in her brain. And when we looked at what these cells were, most were microglial cells and some were oligodendrocytes. And it led us to think maybe we could develop a cell therapy from that cell that could be used to augment a standard transplant. Because one of the problems with transplant is that cells take a month to engraft in the blood, and they probably take 3 to 6 months to engraft in the brain. We don't have a direct way to study it, but it's not right away. And that means in these progressive diseases that the kids have progression of damage before they stabilize. So through many years, and I'll tell you the story, we were able to grow this cell we call DUOC, Duke O cell. One of our patients named it an O cell, version 1. It, over time-lapse photography that Pammy Nogler did in our lab, takes on many different physical shapes and phenotypes, but it's really all the same cell. And it can also crawl across plastic. It divides every 2 or 3 days, so it's slowly dividing, and we've developed it into a cell therapy. Now, in order to do that and be compliant with all the regulations that you need to be compliant with in the days of doing cell therapy today, we had to form a unit that luckily was funded by the Robertson Foundation and is called CT Squared. And much of it is in this building, many of the people who are involved with this are in the room. But basically, CT Squared is now made up of a research laboratory over in RP2, the ability to do animal models, the cord blood bank, and then the stem cell lab interacts with that. That's our clinical transplant lab on the first floor of this building. A new GMP lab, which was fitted into the 9th floor and has 3 different manufacturing suites. clinical trials unit, and a quality and regulatory unit. And all of these things were necessary to be able to develop cell therapies and conduct clinical trials. So just as an example, I'll use the DOC cells. We had a hypothesis that we based on observations in the clinic in our patients. We knew that we'd had to derive the cell and then develop the techniques to manufacture it in a GMP environment, validate that manufacturing, submit an IND, which turned out to be an interactive process with the FDA over a couple of years, to study safety and tumorigenicity, determine mechanisms of action, determine biodistribution, develop release criteria, because I didn't say it, but we're giving this cell intrathecally or into spinal fluid, which means we had to make sure it was stable in a solution that could go into spinal fluid in humans, make sure it was stable in that environment, develop a clinical protocol, submit and maintain an IND, submit and maintain the IRB, and then monitor the study. So quickly, the history of this is that Lisa Tracy in 2008 and 2011, Lisa was a pediatric surgical resident here, and she came into our lab for 2 years and worked on deriving the DOC from cord blood. And she's now pediatric surgical attending here at Duke, so it's kind of a nice story. But she developed and published the first couple of papers about deriving it from frozen and fresh cord blood. And then just this month, a paper that Andy Balber and I and the group at RP2 worked on was published in Cytotherapy, really describing the manufacturing of the cell. But we learned that the cell is mostly microglial. It expresses CD45 and then different macrophage markers, as well as sometimes some oligodendrocyte markers. I'll show you it secretes certain cytokines. It's phagocytic, changes morphology as I showed you. It proliferates. And Arjun Saha has a poster and a paper about to be submitted, and I'll show you the data showing that it also induces myelination in the brain when the brain is damaged. So Bob Storms did the work showing biodistribution, and when we gave the cells intrathecally to 1-day-old mice, we showed that, and these are immune deficient mice, that we could detect cells in the brain and the spine out to 56 days. We didn't go further than that just because the FDA didn't require us to. So some of this was driven by what we needed to do to get the IND. We showed, working with David Wenger, who has a laboratory at Jefferson Medical College and does all the lysosomal disease testing for human subjects, that when we manufactured different lots of the cells, that we didn't alter their ability to produce the lysosomal enzymes, which is part of the way the cells are going to be therapeutic. We showed that the cells constitutively and with stimulation with TNF-alpha predominantly made the cytokines IL-6 and IL-10. IL-10 is immunosuppressive, and we consider that a good thing because they're going into an environment of an inflamed brain in these children with these leukodystrophies. IL-6 is pro-neurogenic. We also considered that to be good, but can be pro or anti-inflammatory. Although in preclinical models it looked safe, and we took it into clinical trials with that knowledge. And then Arjun and Andy developed a model where using cuprizone, which is a copper chelator, which you can feed to mice and then see demyelination of the corpus callosum, which is reversible on a certain schedule, but which we hope the DUOC cells would accelerate. And so basically the mice, and we adapted this to a Hodgkin gamma mouse, were given the diet for 5 weeks, and then they went back on a normal diet, and then the next day they were either injected with DUOC cells into the brain or a sham injection with Ringer's lactate. And then we harvested their brains a week later. And I'll show you a few slides illustrating the effect we saw. So basically this is on the upper panel. The blue is Lexol blue staining of the corpus callosum, which is a big bundle of nerves in the middle of the rat's brain. And normally it looks like the top panel in low and high power. The mice that were given cuprizone, you can see the blue is disappearing and they're demyelinating. The mice that got sham injection with ringers did not remyelinate, but the mice that got intracranial injection with the DUOC did remyelinate at a week, and you can see that graphically presented here. This is another way to look at it. These are tiled confocal sections of the whole brain of the same mice, and or same litter, same treatment, not exactly the same mice. And this is myelin basic protein staining, which stains myelin green. So you can see the control has a lot of green. The cuprizone fed loses most of the myelin. The ringers rescued did not remyelinate, but the DUOC rescued mice did remyelinate. And they don't just remyelinate at the site of the injection, they remyelinate the whole brain. So that's important for a systemic response, and this shows you just at much higher power, the ringers injected versus the cell injected remyelination. We looked at it on electron microscopy, and again, this is the cuprizone treated on the left, where the rings of myelin are severely damaged, compared to the DUOC on the right, where you can see obvious remyelination. And looking at higher power, Arjun looked at just the configuration of the myelin. It established itself in rings, and we were able to see that the number of turns per axon were normal, and that the configuration of the myelin was normal. This is important because some groups have shown certain. I have an avallogenate ring, sorry. Some groups have shown that when you remyelinate the brain with gene therapy, you actually get more myelin, but the configuration of the myelin is abnormal, and the behavior in animals that have abnormally configuration of myelin do not correct. Andy's group was also able to show that in addition to remyelinating, the DUOC cells promoted oligodendrocyte proliferation. So this is not the cells themselves turning into oligodendrocytes. This is stimulating endogenous cells in the brain that are there to proliferate. And again, in Krabbe and other diseases, the oligodendrocytes die. So if we can rescue brain by getting them to proliferate, that will be beneficial for the patient. And finally, Bob showed that with intrathecal injection of labeled cells, so these are DUOC cells labeled with CFSC, which is a fluorescein dye, before they were injected intrathecally, 1 week later when he sacrificed the animals and sectioned the brain, he could see DUOC cells. This is high power in brain parenchyma. They're co-stained. The red is a human nuclear antigen antibody, so that you know that we're not staining an artifact. But this is very important because it means through the intrathecal route we were able to penetrate brain parenchyma, which is what we want to do in the patient. We think that the DUOC cells work by several mechanisms of action. They can clean up debris because they are phagocytic. They secrete the cytokines I mentioned, which can modulate inflammation, and they drive oligodendrocyte development and myelination. And Andy's group is just starting to look at the genes that are expressed in these cells compared to other non-differentiated cells, and they've identified a panel of both developmental and cytoskeletal genes that they think are important. So I'm going to show you in a minute some other ongoing studies we have in our lab that relate to brain injury. But so far we've shown that, Andy's group has shown that DUOC cells actually originate from the CD14 cells in the fresh or the immediately thawed cord blood. But when we compare both genotypically and functionally CD14 cells to manufactured DUOC cells, which takes 21 days, they don't behave the same and they don't have the same gene arrays. So we think we have two different products, which I'll talk about in a minute, that have two different therapeutic indications. And I could have said this backwards, but the reason we know that the DUOC cells come from the CD14 cells is because we did a series of separations and fractionations of whole cord blood and showed that when we depleted CD14 cells, we could not grow DUOC, but when we left or isolated CD14 cells in various cultures, we could. These cells don't grow from the CD34 fraction or a T or B cell fraction, so it really is localized to either mononuclear cells from cord or 14 cells. So all of that brought us to a clinical trial that is opened in February, is staged to enroll a child for the first 3 patients every 2 months, where we're giving the DUOC cells intrathecally. And cord blood is commonly stored in a 2-compartment bag, and it enabled us to, from the same cord blood unit, do the standard transplant using the big portion, which is an 80% of the cell fraction, and give a standard cord blood transplant after chemotherapy to a baby or a young child with one of these diseases. And a week out, from the smaller fraction, which is the 20% fraction, we manufacture the DUOC cells for 21 days up in the GMP lab on the 9th floor. Tracy Gentry leads that group. And after 21 days, the cells grow, and if the patient is healthy and not sick with GVHD and engrafted with the donor cells, which should occur by that time, we inject the DUOC cells intrathecally or in the spinal fluid to the baby. We're hoping that this therapy will essentially provide a bridge till the cells from the systemic transplant permanently engraft in the brain and provide permanent correction. And because these are such rare disorders, we can't really do a randomized trial to study the effects of this. Right now we're studying safety, but when we get to phase 2 or 3, we're going to have to use historical controls to see if we stabilize disease more quickly when we apply this therapy. I'll say also that right now we're using it in the context of a standard transplant because the FDA was concerned that there would be an alloimmunization effect and maybe more inflammation in the brain if we just gave donor cells without any tolerization. But we do hope in the future to be able to treat other diseases that are demyelinating like MS or other adult conditions without having to do a standard myeloablative transplant. I'll also mention, this is the second patient on the trial, that the best thing we can do for these babies with Krabbe is to identify them in utero and to deliver them early and get them to transplant as quick as possible. So to get to transplant, they have to have a workup, a donor identified, they have to have a central line placed. This baby also has a G-tube for feeding, and they have to get 9 days of chemotherapy. So even if you started the day they're born, it's usually 2 weeks or so, 2.5 weeks before you actually do the transplant, which is what gets cells into their body to start correction. So it's a pretty complicated set of circumstances to orchestrate. And you might ask me, well, what about doing something even before they're born? And we do have a protocol for first trimester in utero transplant without chemotherapy, but we have not actually treated a baby yet. We've screened several, but haven't had an affected candidate. So from there, we moved to treating brain injury. And we reasoned that since we knew donor cells engrafted in the brain after chemotherapy in kids with genetic diseases, maybe cells would go to the brain in patients with brain injury. And if we used their own cells or autologous cells, we wouldn't have to give chemotherapy because they wouldn't reject their own cells. So we started on a program that I'll tell you a little bit about, where we have a series of studies using autologous cord blood in the diseases I'm listing. We first showed safety, and then we have a study in babies with hypoxic injury called HIE, congenital hydrocephalus, hypoplastic left heart syndrome, where babies go on ECMO, which can damage the brain, cerebral palsy, and autism. And I'll just show you a little bit about these studies. First, Jessica Sun reported a series of patients we treated, mostly children with cerebral palsy, with autologous cord blood infusions, no chemotherapy, in the clinic through a peripheral IV. And we basically reported safety and quality of these units. And in 184 patients who were about a median of 2 years, we saw very few complications. We had a couple of kids with hives or wheezing, nothing that was unmanageable. And basically this was well tolerated in the clinic. We did have one mother who developed hives when her child breathed the DMSO on her skin, and she had a topical reaction to the DMSO, but she recovered with some Benadryl. That's kind of a unique pediatric experience. But we also learned, even though we learned this was safe, that we couldn't really assess efficacy. all the families told us things like, it's a miracle, my child's doing whatever they didn't do before. I would tell people when I started their IV that it's going to take months to see something if we could see something. But the majority of families wrote back or called within a day or two saying, I know you said we won't see anything, but we already see this or that. And so we decided that in order to really assess whether this is efficacious, we would have to do a randomized placebo-controlled trial. And I'll come to that later. In the interim, though, Jessica did another study, which is still ongoing, with patients with congenital hydrocephalus, where we reasoned that there was a crush injury to the brain from the increased pressure in utero. And we also had access to these babies early and could arrange for optimal cord blood collection, which meant big units that could provide multiple doses. And so we've treated 72 babies. This paper just got accepted in Pediatric research. But 72 patients with 132 infusions, all very young, with various types of congenital hydrocephalus. And this just shows you that 72 patients got an infusion, 40 got 2, 16 got 3, and 4 got 4 infusions. And again, this was all their own cord blood divided into various aliquots or refrozen after thaw into additional aliquots. And it was well tolerated. There were no immune effects. There were no increased reactions with multiple dosing. And this is important to know as we stage ourselves to go forward. We don't know if this is efficacious yet. Jessica is doing a natural history study to figure that out. But our impression is, and the neurosurgeon's impressions are, that these babies are doing better than they would have been predicted to do without any treatment. We're also studying whether or not there's a role for autologous cord blood infusions in patients with autism, and this is in collaboration with Jerry Dawson and her group in psychiatry. And we have an ongoing study for children with autistic spectrum disorders who are 2 to 6 years of age with an IQ over 35, getting a single cord blood infusion with detailed follow-up. And it's really an endpoint and safety study, endpoint finding and safety study. Autism and some of these other diseases are incredibly difficult to study. You know, I got trained as a cancer doctor, and with cancer, you give the therapy and the cancer either is cured or it relapses, and you have very clear endpoints. But with autism or cerebral palsy, the endpoints are much softer because children make some developmental progress on their own as part of their natural history. They respond to certain supportive care interventions, and the measures that you use to assess progress are not as clear-cut. So for autism, we felt like it was very important to figure out what the endpoints could be for a bigger study before we went and embarked on a phase 2 randomized trial. And we also were confident but not 100% sure that there wouldn't be some toxicity of an infusion of cells in an autistic child that we might not see in a child with a different condition. So this study, which is 25 patients, is enrolled, and the children are having 6-month follow-up now. And we're looking at various both reporting and testing and neurophysiologic outcomes to define the endpoints for the next study. And there are also, in Jerry's group, are some scientists who developed the Shank3 murine model of autism. So it's a genetic model, but we are testing human cord blood in that model. And our plans with autism, at least today, this gets changed pretty frequently, is that our next study will be a study where children are evaluated as their cord blood unit is evaluated, and they're randomized, and they'll either get treated with placebo or best donor source, meaning if they have autologous cord blood, we'll give it, but if not, we'll give a donor unit. And then later we'll cross them over so that everybody gets treated with a unit of cord blood or placebo. And this is a design we've adopted, which seems to be working for several of these conditions. Long term, we hope to go into a study also with mesenchymal stromal cells, which we are manufacturing now from cord tissue, and we're working out the ways to optimize that for treatment, where we give 1, 2, or 3 doses to see if we can sustain an effect over a longer period of time. And in the longest term, we hope to take these kids to a randomized trial where they either get their own cord blood unit or mesenchymal stromal cells, depending on, because we don't expect to know which one might be better until we do a randomized trial. Now, we also have been working in hypoxic injury, and the group in Andy's lab with a group of postdocs and Arjun have worked out a brain slice model where we could study some effects in vitro, and essentially this is just a picture of how they make the brain slices, which they put onto a slide, and then we put cells over the brain slice and assess effects of various things. So in this particular set of experiments I'll show you, the brain slice is established, and after 11 days it's exposed to 45 minutes of oxygen glucose deprivation. This is a way to induce a hypoxic injury in vitro, and then it's restored to normal conditions, and then cells are given or not to the tissue by being placed on top in the media over the slice, and then we assess impact by looking at histology and other markers after 3 days. Sachit Patel made these slides, but these are very nice systems where the neurons stain green, the astrocytes stain pink, and the oligodendrocytes stain green. And this is what a normal section would look like. If you induce OGD, there's a huge astrocytosis and loss of neurons, so less green. But if you put cord blood CD14 cells in the culture, you can see that you don't see the degree of astrocytosis and the neurons don't die. So this is the way we use this model to assess the effects of various things. And we're able to show that the main cells that are killed in this type of therapy are the neurons, and the main cells that proliferate are the astrocytes. We then looked at which populations in the culture would protect the neurons from OGD, and we found that it was the CD14 cells or the cord blood mononuclear cells. So being low is good, that means less killing. Being high means a lot of killing. So without any treatment is the red line where everything is killed, and then this is rescue by different cord blood cells. So the mononuclear cells without T cells rescue, or without B cells rescue, but when you take out the CD14 cells, you lose that effect. And when you look at CD14 cells or mononuclear cells from peripheral blood, you do not see the same effect. So there's something different about cord blood cells than peripheral blood. And this just shows you that in addition to the cells having an effect in purple, if you just take the conditioned media from cells that grow and put that on these cultures, you get partial rescue. So there's probably some soluble factors responsible for this as well as contact. Now we took that knowledge into a plan for a trial in children with babies with hypoxic ischemic encephalopathy and children with cerebral palsy, and we worked with a guy named Sid Tan, as well as the neonatologist here. Sid is in the University of Chicago, and he has a baby rabbit model of hypoxic injury, where he essentially prenatally ligates the uterine artery for 40 minutes and induces a hypoxic injury, allows the pups to go to term, delivers them by C-section. They're all born with various degrees of cerebral palsy. And then we gave him human umbilical cord blood cells to give to these rabbits, scanned them on day 1, and then did functional studies on days 5 and 11, which are similar to 1 and 2 years of life in humans. I forgot to put the MRI slide in, but we did show at 24 hours that cells went to the brain. And then in the functional studies, the dark line is the treated animals, the dotted line is the animals getting sham control. These animals got IV human cord blood. These are not immune deficient animals, these are immune competent animals. But what you could see is that the treated animals had better posture, better ability to walk, better balance, and did not develop increased tone or dystonia, which would be signs of spasticity, which is what you see in cerebral palsy. So there was a signal in this animal model that the human cells had efficacy. We then went into babies with hypoxic ischemic encephalopathy. This is where newborns are born with a hypoxic insult after a normal term pregnancy. And Ron Goldberg and Mike Cotton and Amy Murtha have been our main collaborators here at Duke with this study. The first results were published last year in the Journal of Pediatrics. Basically, we have term babies with HIE where there is well-established diagnostic criteria for severity. They have to have moderate to severe encephalopathy and be eligible for cooling, which is the standard of care treatment, 3 days of cooling. And then if we've collected their autologous cord blood and if the parents agree to cooling and the study, the babies are also given autologous cord blood infusions, not banked cord blood, but fresh cord blood at 24 and 48 hours of age, and then they're followed for survival and functional outcomes at 1 and 2 years of life. I'm just showing this to show you these are very insulted babies. Their APGARs have to be less than 5 at 10 minutes. Their pH has to be less than 7, and they have to have a big base excess. So these are our standard criteria established by the neonatal network for this disorder. When the baby is born, the mom, per the IRB, can be asked through verbal assent if it's okay for the OB to collect her baby's cord blood. And if she says yes, the OB collects, and then later it's processed in the laboratory into a small volume in a special bag I'm showing up here so that it can be distributed to the baby. This machine is the same machine we use to process regular cord blood for banking, but instead of cryopreserving it at the end, in this case, we adjust cell dose and hematocrit and put it in a syringe for infusion to the baby. And it's given to the baby over 15 minutes intravenously. Working with the OBs, we learned mechanisms to be sure we captured cord blood collections on all the babies in red that were affected with HIE in the last few years. But to do that, we had to say to the OBs, or they said to us, we're not sure about every baby, you know, what if we collect on a baby who turns out not to have HIE, is that okay? And we said yes. And so blue line is actually the number of collections they've done compared to the number of babies we've treated. And if the baby is not sick in the end of the day or recovers, then the cord blood is discarded at this point. But we have not missed any collections. And we also have worked with referring centers and put collection bags in every delivery room so the OB could just grab it and do this collection if needed. So this just shows you characteristics of babies that have been treated. Now there have been 45 treated at Duke in the last few years. And originally this project was supported through the CTSA and then later by the Robertson Foundation. But at the same time, there were 154 babies who were treated at Duke for the same condition but did not receive cells for one reason or another. And we cheated in the sense that we're comparing outcomes in these two groups of babies that were not randomized but were concomitantly treated. This is just to show severity of HIE in both groups were the same. We treated babies where as little as 3 mils of cord blood was collected. In half the babies, we had enough if we wanted to give 4 infusions, even though we only gave 2. And we adjusted the infusion volume and the red cell content and the cell dose to the baby's weight. This shows you hospital outcomes where between these two groups, there was a trend towards less death in the babies who got cells, but that's not a statistically significant p-value. But in outcomes looking at survival with normal function, which is measured by 3 Baileys, which is a pediatric developmental score that have to be above 85 at a year, the babies in the cell group who got cooled with cells had better outcomes than the babies with cooling alone. Now this is not randomized and we can't take it any further than an observation, but it was enough for us to convince both the Robertson Foundation to fund a multi-center phase 2 randomized trial and also the Neonatal Network, which is an NIH-sponsored network, to agree to do a phase 3 trial if the phase 2 shows a signal. So that's how we'll take it up through the ranks and perhaps get it established as a standard of care. So we are just this summer going to initiate a randomized phase 2 with 8 centers using the red cells from the cord blood as a placebo. So the babies will be randomized to get their mononuclear cells or their red cells from the cord blood. They won't be exposed to anything that's foreign to their body. And then, as I mentioned, if the phase 2, which will take a couple of years to execute, shows a signal, we'll go to phase 3 through the Neonatal Network. Then I'm just going to end talking about a few other things we're doing. We have a randomized placebo-controlled trial going in cerebral palsy. It's being analyzed now. but it was the way that we decided we had to assess whether we saw an efficacy signal in these kids. So you can see that the kids are evaluated, their cord blood is evaluated, they're randomized, and they get an infusion blinded of either cord blood or placebo, which is pink tissue culture media with DMSO. So everybody gets pink stuff and everybody smells, because everybody guesses what they got. And then they come back in a year and cross over and get whatever they didn't get the first time. And the analysis point is first year, and we're using a scale called the GMFM, which is a motor scale that predicts for children with cerebral palsy based on their degree of disability, which is divided into levels, where they will be in a year based on their function at study entry. So we don't have that data, but we will soon. And Alan Song is at the same time looking at connectivity in the brain of the children, both on the CP study and the autism study. So these are connections that are known to be present in the brain. They've been mapped out from babies up to 82-year-olds. These are comparing baseline and 1 year scans in overlays. So red fibers means increased connections, and blue lines means decreased connections. We don't know the treatment of these kids, but we do know that the kids on the top with the increased red fibers all met the endpoint and had improvement, whereas the kids on the bottom did not. So at least we have a biomarker that helps us quantitate neuroconductivity. And just to end, if any or some of these therapies show benefit, we know that many, many people, and certainly adults, are not going to have their own cord blood. And we really want to figure out a way to use banked donor cord blood without chemotherapy to treat some of these conditions. So that's the next thing that we're focusing on in our group. We're hoping that the therapeutic effects are through paracrine signaling, not through engraftment. And we know that timing of the injury may matter as well. So we're trying to sort some of that out in animal models and some of that in people. We're also looking at the safety as a start of allogeneic cells, and we have to start in adults per FDA guidance, and we're doing a study in adults with stroke. So this is opening at Duke, Brady Hospital, and MD Anderson, and it's adult patients with ischemic stroke who can be treated between 3 and 10 days post-stroke with a non-HLA matched, off-the-shelf, banked public cord blood unit that is matched for blood type and race. And we'll look at safety and then a randomized study for efficacy. Daniel Laskowitz did this work in his animal model of stroke. It's a murine model with an MCO infarct. And we just showed that infarct volume was smaller if the animals got cord blood at 5 days, that survival was better if the animals got cord blood, and that their neurofunctional scores were improved if they got cord blood. So this is kind of our plan. We're doing a phase 1 now, and then we're hoping to go to a phase 2 next year, which will be randomized between, again, placebo and an IV dose of cord blood. So I'm going to stop there and just again say, Hillary Clinton said it takes a village, but it really does. I'm the vehicle of explaining the work we're doing to you, but I work with literally a few hundred people, many of which have been with me for more than a couple of decades, who've been dedicated to all the aspects of doing this work, from the clinical team who takes care of the patients to the stem cell lab to the cord blood bank to our research group and our collaborators. And this work has been funded by multiple sources, some from the NIH, the Julian Robertson Foundation, and the Marcus Foundation, which have really given us big grants to support the clinical trials I described. So I'll stop there and see if you have any questions. Thank you.
Remarkable work. Thank you so much. Time for a few questions.
Oh, you can't not.
Yeah.
I wanted to make a comment, but I'm sad that Eric Peterson left. I think, Joanne, if you would just help us understand what we've learned by being asked by FDA to go back into animals to demonstrate some of the things that we had to demonstrate. You know, those of us who do clinical research and translational research aren't mad about looking at mouse data, but it might shock you to know that some of the mouse experiments that we did in order to demonstrate things that the FDA wanted us to be able to demonstrate. So it's really like back translation, and I wonder if you would speak to that for just a second, or maybe Amanda too.
Well, I'll start. I think we're in a little bit of an unusual situation in that we didn't follow the traditional route to get to where we are. So because we were doing standard of care bone marrow cord blood transplantation, which was not regulated in the way that cell therapy is, we had already treated patients and made observation in patients that we couldn't ignore as far as I'm concerned, and I think may have fast-tracked us a little bit forward. But I think when you are using novel cell therapies or manipulated cells that are not de novo cells, just harvested from a donor, there are safety issues that you can address in animals to some degree. I think the efficacy issues are still a controversial conversation. FDA requires it. I'm not sure it's all that helpful. I just came back from a meeting where people are talking about, thousands of MSC experiments in stroke and other diseases that look great in animals, and as soon as they modeled it in people, it didn't do anything. So we've had the same experience with ex vivo expansion of cord blood. We can expand cord blood 800-fold. We put it in people and it doesn't do anything. So I think the efficacy side is more difficult to address, but I learned the hard way that we had to do what they wanted in order to get to the clinic, so we did. And we had to put together a huge infrastructure to make that happen.
I had a non-scientific question, actually, but as many of these diseases and these children have to be treated immediately after birth or very soon after birth, what is in place for the support and financial backing for these families?
So usually the medical care is covered either by Medicaid or third-party payers, private third-party payers. And we've established precedent over the years for these very rare disorders to make that happen. In our PEDS transplant program, we have 2 dedicated financial analysts who just work to get insurance approval and make sure that the medical stuff is covered. But for all the other costs of moving away from home and, still having to pay rent or take care of your other kids or taking time off from jobs, we have multiple ways to try to help families. We have a family support program that we established 15 years ago that's fully supported through philanthropy that helps with support groups, with financial support and resource support for all the different needs that families have. And also with just making things more fun for the kids than just being in the hospital. We have hospital school, which is from the Durham public school system, but really makes a difference for the kids. But it's sort of a bucket we can never completely fill. So we always have more needs than we can meet, to be honest. Some of our families have even bought townhouses and make them available to other families who are coming to stay here. So there have been a lot of pay it forward and giving back, but it's a grassroots kind of effort.
Real quick.
Most of the stuff you talked about today is genetic. Are you guys going to be branching off, or do you have a section that you're focusing on traumatic injuries? I know Danny Laskowitz does a lot with the brain.
Yeah, so we're thinking about that. It's harder to model a study in TBI because it's very varied. We actually talked with WOMAC, the Army base, to try to figure out a way to recruit and enroll patients into that kind of study. We're putting the various types of cells we're working with into Danny's model. And the first couple we put in didn't really show any efficacy in the mouse. I don't know if that means we should or shouldn't pursue it, but we're moving forward in that direction. Yeah.
One quick question. So looking back to 1983 with that early experience, could you have imagined coming this far 3 decades later? And then
No.
the second part is, what's in store 5-10 years from now? This is really transformative work that's pretty remarkable.
Yeah, so the answer to the first question is no. Didn't really know where this was. If you asked me then, where would I be, I wouldn't have said that. But of course, in college, there was this essay, Imagine Yourself at 35, and not for God knows why, but I said I wouldn't be a doctor. I still remember that. But anyway, but as far as where do I think this is going, I think regenerative medicine, cell therapy is the next emerging important field in medicine. I think these cells are factories of drugs that we don't know how to make, and that they can exert effects that we can't harness another way right now. And that that's really going to be a very important development in medical therapy, both for injury, stroke, heart disease, et cetera. But I think it's going to take a good 10 to 15 years to have something be routine in the clinic.
Well, thank you for ignoring what you said in your college essay and for your presentation today.